Hello, everybody. It's Mr. Caudell. Let's finish chapter 26. I'm going to tell you, I'm not going to lecture on 26.4 or 26.5. The title of 26.4 in the 10th edition is An Organism's Evolutionary History is Documented in its Genome. That makes sense to me that you could look and see where genetic mutations have occurred or changes over time, which is going to lead to different characteristics. Which is going to lead to characteristics that are selected for in the environment, either positively or negatively. And then you're going to have evolution over time. I think it's good. There's nothing for me to really lecture on here. And then I like, then you could use that kind of consistent change of DNA that I've mentioned a few times now throughout time in order to use it as a clock. And so then when someone says, oh, we diverged from a rat 37 million years ago from our common ancestor with a rat, the point is that we can go back and say that number because we know the consistent rates of DNA change. So I think it's 26.4 and 5 are super important. Make sure that you read them. They will be on the test for sure. There's just nothing for me to say. I'm going to talk for a few minutes. It's not going to take long. About 26.6. Our understanding of the tree of life continues to change based on new data. I thought that there was a couple things to mention here. But of course, let's begin with the duck-billed platypus. Where would you put it on a phylogenetic tree?
Settlers thought it a hose. Duck-billed, otter feet, beaver tail. Eastern Australian mammal that lays eggs. Monotrees. Electroreceptors on bill. Senses prey's electrical current. Hidden mouth under bill. Eats gravel. Eats insect larvae and freshwater shrimp. Only eats on land. Stores prey in pouches. Eats 20 to 30% of body weight daily. 12 hours a day on food. Feet wet. Walks on knuckles. 30% more energy used on land versus water. Loses teeth early in life. Grinds food. Rough pads in mouths. No stomach. Esophagus leads straight to intestine. Male platypus is venomous. Ankle spurs on back legs. Venom can kill small dogs.
Well, I try to make an argument to you that everything is stepwise and linear, and you can draw these trees, and you can make the next outgroup because it doesn't have the next characteristic in the evolutionary of that monophyletic clade as it goes through time. Well, look at this thing. This thing has venom. It lays eggs like a bird, and then it lactates or produces milk like a mammal, and it does all those other things. No one knows where to put this thing on the evolutionary tree, and it's not alone. Spiny anteaters, we don't know where to put them on the tree either. They're mammals which lay eggs. Now, I thought that the definition of mammal was live young, produces milk, has hair. Okay. Also, there's an oriental hornet here. It's an insect that lives, and it photosynthesizes. Yay. Also, I read in the book that aphids, which are these little insects that suck kind of juices out of plants, they have carotenoids, which are also photosynthetic compounds which absorb certain wavelengths of light. The point is that new information continues to revise our understanding of the tree of life. Again, I gave you the basics, but now I'm about to throw all that away and say, have an open mind. Basically, and so there's evidence of this in phylogeny or this practice of creating these trees for certain scientists that do this. Over time, we've gone from 2 kingdoms to 3 domains. At the beginning, it was either plants or animals for the first phylogeneticists that did this. We've obviously amended this over time, and you can only imagine all the kicking and screaming that happens when someone proposes an amendment to this stuff. It's like, I have a new idea, let's change the way we classify organisms, and the whole biological community goes nuts. So there were 5 kingdoms, the Monera, which are the prokaryotes, the protists, which are single-celled eukaryotic organisms like amoeba that live in lakes and puddles and stuff, plants, fungus, and animals, no problem. More recently, and this was just in the last 10 to 15 years, the three-domain system has been adopted, and this was due, as I've said before, because there were these single-celled organisms that lived in weird environments like salt flats of Utah or under the ice in Antarctica or in boiling hot pots in the Yellowstone National Park, and people said, those are just bacteria. Now that, we've been able to sequence their genomes, we realize that they have more in common with eukaryotic cells than they do with prokaryotic cells. So they're even further from bacteria than we even thought that they were when we just used to lump them in with bacteria when I was a youngster. So the point is we now have a three-domain system. And that's when you go down here and that mnemonic for remembering the phylogenetic taxonomic classifications, that's why we have dumb King Philip came over for good soup, because we needed the D for domain. This is supported by data from many sequenced genomes.
A few key things to note about the three-domain classification system. What's a domain? It is a group within the classification system, or a taxa. It is the most general taxa. All life fits into 3 domains. Like a superkingdom, each domain includes 1 or more kingdoms. Bacteria has its own kingdom, and Archaea or Archaeobacteria also has its own kingdom. And then all eukaryotic organisms are organized into also 1 domain.
I tried. Oh, that's the best I could do. So here's how your book organizes them whenever your book was published. So we've got the domain eukarya up there at the top. These are the organisms with eukaryotic cells, and you can see how they're related to one another. It includes animals, and then these things like algae and ciliates and these single-celled organisms that are way up there at the top, or I guess all the way from amoebas up to the top. And then you've got these archaeobacteria here there in the middle, and then the bacteria way down there at the bottom. So here's the eukaryotic organisms. Here how they're related. Here's the archaeobacteria. I have a little movie here that'll show you a little bit more about these very interesting organisms on our planet.
We've all seen movies about terrible insects from outer space, or stories of abduction by little green men. But the study of life in the universe, including the possibility of extraterrestrial life, is also a serious scientific pursuit. Astrobiology draws on diverse fields, such as physics, biology, astronomy, and geology, to study how life was formed on Earth, how it could form elsewhere, and how we might detect it. Many ancient religions described other worlds inhabited by non-human beings, but these were more like mythical realms or parallel universes than other planets existing in the same physical world. It is only within the last century that scientists have been able to seriously undertake the search for extraterrestrial life. We know that at the most basic level, organisms on Earth need 3 things: liquid water, a source of energy, and organic carbon-based material. We also know that the Earth is just the right distance from the Sun, so as not to be either frozen or molten. So planets within such a habitable range from their own stars may be able to support life. But while we used to think that life could only exist in such Earth-like environments, one of the most amazing discoveries of astrobiology has been just how versatile life is. We now know that life can thrive in some of the most extreme environments that would be fatal for most known organisms. Life is found everywhere, from black smoker hydrothermal vents in the dark depths of Earth's oceans, to bubbling hot acidic springs on the flanks of volcanoes, to high up in the atmosphere. Organisms that live in these challenging environments are called extremophiles, and they can survive at extremes of temperature, pressure, and radiation, as well as salinity, acidity, and limited availability of sunlight, water, or oxygen. What is most remarkable about these extremophiles is that they are found thriving in environments that mimic those on alien worlds. One of the most important of these worlds is our red and dusty neighbour, Mars. Today, astrobiologists are exploring places where life might once have existed on Mars using NASA's Curiosity rover. One of these is Gale Crater, an impact crater created when a meteor hit the surface of Mars nearly 3.8 billion years ago. Evidence from orbit suggests past traces of water, which means the crater might once have supported life. Planets are not the only places that astrobiologists are looking at. For example, Europa, one of the moons of Jupiter, And Enceladus and Titan, 2 of Saturn's moons, are all exciting possibilities. Although these moons are extremely cold and 2 are covered in thick ice, there is evidence of liquid oceans beneath this shell. Could life be floating around these oceans? Or could it be living around black smoke events at their bottom? Titan is particularly promising, as it has an atmosphere and Earth-like lakes, seas, and rivers flowing across its surface. It is very cold, however, too cold for liquid water, so these rivers may instead be flowing with liquid hydrocarbons such as methane and ethane. These are composed of hydrogen and, more importantly, carbon, which is the basic building block of all life as we know it. So could life be found in these lakes? Although instruments are being designed to study these distant worlds, it takes many years to build them, and even longer to get them where they need to be. In the meantime, astrobiologists work in our own natural laboratory, the Earth, to learn about all the weird and wonderful forms of life that can exist, and to help us one day answer one of humanity's oldest questions. Are we alone?
All right, so here's the last stuff of the bacteria. We are going to study a little bit of chapter 27, which is the next chapter, and it has to do with prokaryotes, or the domain Monera, which are the bacteria. Don't need to know too much about it right now. I'll just take a look at it in the textbook for this chapter. Well, if we're going to make a huge, big tree of life, we would use rRNA genes because these have evolved slowly. Remember, these are the genes that create RNA or rRNA, or type of RNA, that then will go into the nucleolus combined with proteins to create ribosomes that do translation out in the cytoplasm, either bound to endoplasmic reticulum or not. So these are just what scientists use. What they have found when they look at rRNA genes across species is that they found this thing called horizontal gene transfer. And they found it in birds, plants, humans, and even these geckos. I think it says in the book that there's 17 reptile species that have acquired a gene that creates a certain type of rRNA, but it's found in something that's totally unrelated to a gecko, for instance. So there's no way, it wasn't a linear, it's almost like the platypus example. It wasn't a linear progression that you could see stepwise through time. It's kind of like this circling back of information and that has now created characteristics and has been put in organisms that you wouldn't think it would be in. So here's my point, is that if this is what the old view of the tree of life looked like, here's our common ancestor, and here's all this branching superstructure of evolutionary relatedness or unrelatedness. Now, basically, I'm just going to put some lines here. And what these lines are to represent is the transfer of genes from organisms that you wouldn't expect to have genes transferred between. And when this all really went down, this is what your book says, but I can't explain the oriental hornet or the platypus or the spiny anteater or the gecko. When this really all went down was way back in time. If you, again, have the x-axis as time here and you're going back to the left, you can imagine that when things were single-celled and they were just cruising around in the primordial soup, whether or not they were organelle-like, like I'm looking at the picture, like a mitochondria or a chloroplast, or maybe bigger than that, there was a lot of kind of sharing of information and sharing of stuff, and things were being endocytized and phagocytized all the time and being enveloped. And so this horizontal gene transfer, they think this is when all that really went down. It was a long time ago, over 1 billion years ago. And so is the tree of life that we've been drawing and teaching our students forever, maybe it's just a web of life where there's all this interconnectedness. That's the question. Go ahead and press pause and try to answer these 2 questions out loud. Oh yeah, I found this on my desk today, but I think you're awesome. Thanks for listening.